Hi, Dr. Wang here. Today we're going to do a practice test for Exam3, the bioinformatic component. In the first question, we're going to use gene bank record to figure out whether the following genes are promoter, CDS, intron, or UTRs. To do this, we're going to first open the gene bank record for the genomic DNA. I have this record already open. The next thing is to turn on highlight sequence features. This is going to open a small tab at the very bottom. So currently we can see it's displaying CDS. In this stretch of genomic DNA, there are a total of 5 different CDS or 5 different genes. So the next thing we're going to do is to open up the messenger RNA record. As we can see here, this gene is called UQCC3. So I'm going to flip through the 5 genes to see which one is UQCC3. And as we can see here, the very last one is UQCC3. And we're going to work with this gene. So coming back to the sequences, I'm going to copy the first sequence, come back, and then search for it. So we can see that this sequence is located outside the CDS, right? So for this, I'm going to switch back to the exon view and then go to the first exon. So we can see that this sequence is located outside the first exon, so which means this is a promoter. So the second sequence, all right, so we're going to do the same search. And before doing so, I'm going to go back to the CDS view, make sure we're working with the 5th gene, and then do this search. Oops, I think I copied a little too much. All right. Okay, so we can see that this sequence is located in between, or it's breaking the CDS region apart into 2 parts. So this is the intron sequence. All right, so the next sequence, do the same, copy paste, and then come to here and then search for it. So here we can see the CDS itself is highlighted. So this is a CDS region. Okay, the next sequence, and again, we're going to do a search find, and actually it highlighted CDS again. So this means that this is a CDS as well. Right, so we're moving along to the next sequence, copy paste, and then search for it. So here we can see that it's outside the CDS. This is when we need to switch to the exon view to see where exactly is this located. All right, so here let's search for this, and we can see that it's, even though it's outside the CDS, it is located within the 2nd exon, which is the last exon. So here, this is the 3' UTR. So here, 3' UTR. Okay, so let's do the next search. So copy paste, come back, and we're going to search. So currently we are looking at the 2nd exon of this gene, or the last exon of this gene, right? So, and then it's at the very end. So if I go back to the CDS view, we can see that this gene, let's search again, this sequence is located within the exon, the last exon, but not in CDS. So this is again a 3' UTR. All right, so now let's look at the last sequence. The last sequence, so I'm going to search for it. Right, so here the last sequence is located right here outside the actual CDS. So then I'm going to switch to the exon view. So go to the first exon and then do this search one more time. So we can see that it's located within the first exon, but not in CDS. So this is the 5' UTR. All right, so this is how to solve the first question. So for the second question, we're going to use a different platform called the Genome Browser. Please use the embedded link. So I'm going to click on this link. This opens up the Genome Browser. And within the Genome Browser, I'm going to look for this gene called UQCC. So place gene name into the search box and then simply search. So this is going to give you some result. Usually the first one is what we're looking for. So UQCC. So currently we are looking at this gene on the human genome. So we can see that this gene is on chromosome 20, and this red line represents the location of the gene on chromosome 20, and we can see that this is on the Q arm. So the centromere is right here. This side is the P arm, this side is the Q arm. So another name for the Q arm is the long arm. So here we have chromosome 20, and then, chromosome. So it's located on chromosome 20, and then it's located on the Q arm, which is the long arm of this chromosome. So the last question asked for, is it located on the positive or negative strand? So here, the UQCC gene, we can see that the intron region actually have tiny arrows that's going from right to left. So this means that UQCC gene is on the bottom strand. So it's going from right to left. All right, so coming back, this is the bottom strand. In the previous question, we were looking at the UQCC gene on the genome browser. So this gene, there are a couple of different versions on the genome browser. We are going to use this highlighted version to figure out the answer for question 3. So in question 3, the question gave you coordinate on the genome browser and asking you to figure out the location of that coordinate. So what we're going to do is to copy the coordinate and then come to Genome Browser. So over here, we first need to tell Genome Browser that we are looking at chromosome 20, right? So I'm going to click on this. So this is going to copy chromosome 20 into the search box. And then I'm going to paste the first coordinate into the search box. So make sure you have chromosome 20 in front of it. In the actual exam, since each question is randomly generated, for this question, you will have to search for the presence of the gene first, and then go to that chromosome and then paste the coordinates right here. So I'm going to click go. This will go to exactly this nucleotide, and we can see that this is a T. So over here, the first thing I'm going to do is to zoom out so that we can see the surrounding context. So I'm going to zoom out 100-fold first, and then I'm going to zoom out 10-fold. So it looks like I still need to zoom out. So in total, I zoomed out probably 10,000 times. So over here, we can start to see this exact coordinate 891,000, right? So this 891,000 is right here. So because this gene is on the bottom strand, so it's going from right to left, we are looking at the UTR region. On Genome Browser, the graphic feature for UTR is this relatively short box. The coding region is this relatively tall box. So currently we are looking at the UTR, and it's at the 3' end of the gene. So coming back, this is the 3' UTR. Then we are going to start with the second coordinate. So just replace the first coordinate with the second one and then search. So over here, we can see that this is the intron region. So let's zoom out 100-fold, and I'm going to zoom out another 100-fold. So this coordinate 894,000, okay, it's right here. So we can see that this is intron and it's the last intron, okay? So because this is the 3' end of the gene. So this is the last intron of the gene. All right, so let's move on to the next. The steps are similar. All right, so here we're looking at intron again, right? So once the graphic element looks like, wait, oh, actually this is not the gene. All right, so this is not the UQCC gene. So let's zoom out to see what's going on here. So I'm going to zoom out. Oh, okay, so now we can actually see that. I'm going to zoom out perhaps 10 more fold. Okay, so here we can see this more clearly. So this particular coordinate 999950. Right, so 9999 50. So this is probably somewhere in here. Right, so this is right here. Sorry, so it's fairly close to this landmark right here. So it's in here. We can see that this is the very 5' end of the gene. So this is outside the gene, which means this is the promoter region. All right, so now let's do the next one. Okay, so something is coming up right here. So this looks more like UTR to me. I'm going to zoom out just to be sure. All right, so over here, this coordinate is 9, 5. Oh, okay, so this is actually 902, right? So 902, 520. Oh, this is not UTR. This is actually in the coding region. We can see that this is coding for individual amino acid. So here we are looking at the coding region, CDS. All right. So, and I can zoom out a little bit more so you can see this better, right? So here, it's actually here. This is the coding region. And then let's do the next. All right, so here we can see that we're in an intron region. So I'm going to zoom out to see which intron. Let's zoom out 100-fold. Okay. Okay, so 99000, all right, so it's right here. So we can see that this is the first intron, right? So over here, this is the start of the gene. So this is the first intron. All right, so very last one. All right, so and then we're going to do the same. I'm going to zoom out first. All right, so it looks like I need to zoom out again. Okay, so now we can see this a little bit better, right? So it's 9999900. So over here, it's very close to the 34 landmark, somewhere in here. So for this, I'm going to zoom in a little bit more. Okay, so now we can see this 3399990. It's right here. So this is located in the UTR region. And because this is the very 5' end of the gene, this is the 5' UTR. We can see that the UTR is the shorter box, right? So the taller box over here, this is the CDS. So coming back, this is the UTR region. So for question number 4, we are going to perform a blast. The sequence given to us is protein sequence. So let's copy this sequence and then perform a protein blast. So over here, just simply paste the sequence in the search box and then blast. I have the result that's already here. So this is after blast, we get a series of hits. So usually the first hit is what we're looking for. We can see the name of the gene is right here. And then this is the species name. If you're unfamiliar with the common name, you can simply Google. All right, so over here, the gene name is right here, right? So this is just basically coming from the blast. And then the organism is mouse. Hi, to solve question 5, the first thing we're going to do is to find the gene bank record for this protein. So first, copy the accession number and then go to gene bank. Because this is a protein record, we are going to look for proteins. And so select the option for protein and then paste the accession number into the box. This is going to pull up the gene bank record for this protein and the protein sequences right here. So the next thing we are going to do is to translate the mutant sequence. So over here, I'm first going to copy, this is fairly long, right? So to make sure that you have copied the entire sequence, and then go to Sapio and then paste the sequence right here. All right, so the next thing we're going to do is to look for the longest open reading frame, right? So look for ORF detection. So the sequence that we gave you is basically a linear sequence. And then I'm just going to search for the forward direction because we usually give you the coding strand sequence. So scan. And after scanning, I'm going to minimize this window so I can look at the entire thing, right? So we can see that this green line represents the open reading frame. And there's only one open reading frame discovered, and it looks like occupy the majority of the sequence. So this is the longest open reading frame, right? So I'm going to click on this and then translate the forward direction. All right, so here, this is basically the mutant sequence. So I'm going to copy this sequence and then now open a sequence alignment tab. So this is the mutant sequence, right? So I'm going to put this mutant and then paste the sequence in here. So now I'm going to go to the gene bank record, copy paste the wild type protein sequence into the first box, wild type, and then paste. Right, so here, now we're going to run the sequence alignment, make sure that you select protein and then run analysis. So we can see that the wild type sequence and the mutant sequence lined up mostly, right? So except for this mutation. So this mutation is in position when you put your cursor next to this amino acid, it's going to tell you this is isoleucine 179 mutated to a phenylalanine in the mutant, right? So then the answer for this is basically I179F.